Today's topic is about regulation of gene expression. So, various genetic information present over the DNA, and if that particular gene over the DNA is expressed, then particular protein is synthesized. So, protein synthesis occurs under the influence of the gene expression. Now, sometimes the gene which is not expressing is said to be a state of repression inside the cell. So, before that, some particular genes are called as constitutive genes or otherwise called as housekeeping genes. So, these are the genes which are expressed almost always in all cells. Simple genes which are needed for the production of glycolytic enzymes. So because glycolytic enzymes are present in almost all cells to utilization of the glucose or for the energy production. So these are the called the housekeeping or constitutive genes. Now in detail of this gene expression, before that you have to understand these two terminology, induction and repression. So a gene may be alternatively open and shut as per the metabolic needs of the cell. So if it is open or expressed, that is called induction, and it is shut down or not expressed, then it is called repression condition. So induction, increased synthesis of protein, the gene is expressed, while repression, shut down of protein synthesis, then it is called gene is repressed. Now, based on that, a operon concept is given, which is explaining this gene regulation. Jacob and Monod got Nobel Prize for this operon concept. Cells in glucose medium lack beta galactosidase. Simple to explain this operon concept, one lactose metabolism example is given. So here one cell which is present in glucose medium which is lack of this lactase enzyme. So whatever the cell will utilizing the glucose, in that cell lactase enzyme which is needed for the lactose metabolism, so this lactase enzyme is not present in that cell. But suddenly what happen, cells when it is putted in a lactose medium get 1000-fold level of this enzyme lactase. So same cell which is lacking of this enzyme suddenly get this all lactase enzyme, and how it is explained, how it is occurring, that is explained in this operon concept. So whatever this example is given, and for that it is called the lake operon concept or theory. So to understand that, we have to understand operon is considered a unit of gene expression, and these include control elements. So they are called a regulatory site or gene, promoter site, operator genes or operator site, and one more, that is structural genes. So operon unit is basically made up of these four parts: regulatory site, promoter site, operator site, and structural genes. So in detail of these four that we will look. So over the DNA, that particular ZYA, that is the structural gene which is responsible for the production of enzyme which are needed for the lactose metabolism. Now this gene that is connected with these three, regulatory, promoter, and operator site or the gene. So transcription is regulated by this regulatory gene. Now this regulatory gene is also called the inhibitory or inhibitor gene. So that is basically producing a one repressor molecule, and this repressor molecule binds to the operator site. So basically these act as a hindrance for the RNA polymerase enzyme needed for the transcription of the structural gene. But when this lake repressor which is produced by regulatory gene and attach over the operator side, this RNA polymerase is not able to move over the structural gene. So this will occur in absence of lactose in medium. So simple, lactose utilizing enzyme which is produced by that ZYA gene is not synthesized when this lactose is not present in the medium. So simple, cell does not containing lactose, so there is a one molecule is produced by the regulatory gene which will remain attached over this operator site. So the RNA polymerase is not able to synthesize this structural gene which is utilized for the lactose metabolism. So promoter site, that already we have seen that over that RNA polymerase bind, so transcription of the structural gene will be possible, but that will occur when lactose will be present. Otherwise that RNA polymerase is not able to attach or move further over the structural gene. So basically that structural gene that we have seen in the picture ZYA, so Y gene, that is for the production of permease, that is transporting lactose and galactose into the cell. So Z gene encode for the beta galactosidase hydrolysis lactose to galactose and glucose. Simple, it is lactase enzyme. further a gene that is called a thiogalactosidase transacetylase enzyme. So ZYA gene that is producing a basically enzyme which are responsible for the lactose metabolism. So here it is explained in this picture, glucose is present and lactose is absent. So during that time cell does not produce lactase enzyme. But why this lactase enzyme is not produced, that is explained by this Lake operon concept. So Lake operon that is in a repressed manner. So basically we have seen that there is a four side to explain this operon. You have to remember RPOS. R means regulatory site, P means promoter site, O means operator site, and S means for the structural gene, that is ZYA. Now, when this lactose is absent, during that time we did not need any enzyme for the lactose metabolism. So repressor site or repressor gene will produce a repressor molecule that will attach over the operator site. So once it is present over the operator site, RNA polymerase cannot move further over the operator site to the structural gene. Operator site is generally a short region of about 25 base pair long. So this RNA polymerase cannot move over the structural gene, and this transcription will not occur and enzyme to degrade lactose will not be produced. Now another condition is that de-repression of Lake operon. So what happens when previously lactose is not present, but when lactose is introduced, so now lactose is present. So what happens, lactose will attach over the repressor and lactose repressor molecule complex will be generated that cannot bind to the operator region. Repressor molecule alone that can bind over the operator region, but this complex of lactose repressor is not able to attach over the operator region. So RNA polymerase can transcribe structural gene which are translated to the protein and or the enzyme which are needed for the lactose metabolism. So lactose act as an inducer of this gene. So in the presence of this lactose only, the gene that is ZYA are induced or expressed. So that is explained in this picture. Glucose is not present and lactose is introduced or present in the cell. So what happened, this repressor molecule or repressor site which is responsible for the repressor molecule formation that is produced, but when lactose is present that will attach over the repressor molecule. Now once there is a lactose repressor molecule that is not able to attach over this operator site, so whatever the RNA polymerase attach over the promoter site, that will go further over the structural gene, and this particular structural gene will be transcribed and further translation will occur, and particular protein or enzyme will be synthesized which is needed for the lactose metabolism. So now finally when lactose is present, with the presence of lactose, the enzyme which are needed for lactose metabolism will be synthesized and that will be utilized for the lactose metabolism. So other example of this de-repression or removal of this repression or induction of enzyme, they are the induction of tryptophan pyrrolase and transaminase. So this enzyme will be expressed in the presence of glucocorticoids. Same way, one drug that is, you have to remember, barbiturates. So when barbiturate is present, ALA synthase production will be increased, which is involved in a heme synthesis. So that's all about the Lake operon concept or explaining lactose metabolism. So that is important gene regulation that you have to remember.